Good morning, all. I'm Dr. Latha, Professor of Physiology, Sri Lakshmi Narayana Institute of Medical Sciences, affiliated under Bharat Institute of Higher Education and Research, Pondicherry. Today I'm going to discuss about smell and taste sensations, physiology of smell and taste sensation. First one is olfaction. Olfaction is nothing but sense of smell. Sense of smell is well developed in dogs and rabbits. It gives warning about the environmental dangers to the animals. So that kind of animals are called macrosmotics, and it is when compared with macrosmotics, it is less well developed in humans and primates like monkeys. It helps to the pleasure of the food. It helps in the pleasure of the food. So humans and primates are called microosmotics. So sense of smell, it is mainly present in the olfactory mucous membrane of the nose, which is located in the roof of the nasal cavity. The receptors present in the olfactory mucous membrane are mainly responsible for the sense of smell. Olfactory mucous membrane, it is present in the roof of the nose. It is dull yellowish brown in color and located in the upper one-third of the nasal cavity. The total surface area of the olfactory mucous membrane is 2.5 square centimeter and it is covered by mucus layer. Olfactory mucous membrane is the place in the body where the nervous system is closest to the external world. So the only place where the nervous system is close to the external world. Olfactory mucus, in this picture you can see olfactory mucous membrane or olfactory epithelium. Okay. The olfactory mucosa or olfactory epithelium, it consists of three types of cells: receptor cell, sustentacular cells, and basal cells. The nerves supplied to the olfactory mucosa are mainly by the olfactory nerves and the general nerve supply by the trigeminal nerve. Trigeminal nerve, it is mainly stimulated by the irritant stimuli. Olfactory mucosa, it contains three types of cells: olfactory receptor cells, supporting cells, and basal cells. The olfactory receptor cells, the receptor cells are mainly responsible for the sensation of smell. Olfactory mucous membrane contains 10 to 20 million receptor cells. Each receptor is a bipolar neuron. Each receptor cell is a bipolar neuron. In this picture, you can see it has dendrites and axon. So they are located in the olfactory mucous membrane and also between the supporting cells. If you see the olfactory receptor cells, it has thick dendrite with expanded end is called olfactory rod. In this picture, you can see, so it has a thick dendrite with expanded end. Okay, this part is called olfactory rod. So from this olfactory rod, here you can see from the olfactory rod, many cilia projected to the mucous membrane, olfactory mucous membrane. Each receptor cell has 6 to 12 cilia. This cilia forms the dense mat in the mucus layer. So that only receive the olfactory stimuli. Olfactory neurons have short lifespan of 60 days. Then the new receptor cells are produced by the mitotic activity of the basal cells. The olfactory receptor cell is the only cells very close to the external environment. Next, sustentacular cells are supporting cells. They are present between the receptor cells. They are columnar in shape. Microvilli extend from the surface. Here also, microvilli extend from the surface of the sustentacular cells. These cells that microvilli, it covers the olfactory mucosa. Then this supporting cell, the main role of this supporting cell is secretion of mucus. Mucus also produced by the Bowman's gland placed just under the basal lamina of the olfactory mucous membrane. Third cell is basal cell. Basal cells are stem cells. The main role of this basal cell or basal cells are it replaces the receptor cells by mitosis. When the receptor cells are damaged or after a lifespan, the cells are replaced by the basal cells, mitotic activity of the basal cells. And the special features of the olfactory mucosa, it contains receptor cells, presence of Bowman's gland, yellowish brown in color, absence of rhythmic ciliary beating. Okay, normally in the nose, it has respiratory mucosa and olfactory mucosa. So we can differentiate the olfactory mucosa from the respiratory mucosa by presence of the receptor cells, presence of the Bowman's gland, and the olfactory mucosa is yellowish brown in color, but respiratory mucosa is not yellowish in color. And respiratory mucosa has rhythmic ciliary beating activity, but olfactory mucosa doesn't have rhythmic ciliary beating activity. So next one is olfactory pathway. Olfactory pathway, it is how the sense of the smell is carried to the brain. Okay. The axons of the olfactory receptor neurons. Olfactory receptor cells are bipolar neurons. It has dendrites and axons. The dendrites, it forms a thick rod-like structure with expanded end. So then from that expanded end, many cilia arises. That cilia, it receives the olfactory smell. Okay. That olfactory stimuli, it is transmitted through the axons of the bipolar neuron. The axons of the bipolar neuron, it pierces the, here you can see, it pierces the cribriform plate of the ethmoid bone and enters into the olfactory bulb. Olfactory bulb is nothing but it is a thin strip of the gray matter present on the ethmoid bone. And there is a point-to-point representation of the olfactory mucous membrane in the olfactory bulb. Okay, the anterior part of the okay. So the upper part of the olfactory mucous membrane, it is represented in the anterior part of the olfactory bulb, and lower part of the olfactory mucous membrane, it is represented in the posterior part of the olfactory bulb. So the olfactory bulb, it has many cells, mitral cells, tufted cells, granular cells, and periglomerulus cells. The axons of the olfactory, axons of the receptor cells, it enters into the olfactory bulb where it snaps with the dendrites of the mitral and tufted cells and forms the olfactory glomeruli. So each and mitral and tufted cells, it receives approximately around 1000 olfactory receptor cells. It snaps with 1000 axons of the 1000 receptor cells. And the axons of the mitral and tufted cells, it forms the mitral and tufted cells are the second order neuron. The axons, it forms the olfactory tract. The mitral and tufted cells, it snaps with the granule cells and it stimulates the granule cells. But granule cells and periglomerular cells, which in turn inhibits the mitral and tufted cells. So that is called lateral inhibition. Same thing given in this flow chart. Axons of the olfactory receptor neuron pierces the cribriform plate of ethmoid bone and enters into the olfactory bulb, where it snaps with the dendrites of mitral cells and tufted cells and forms the olfactory glomeruli. Periglomerular cells are inhibitory neurons that connecting the one glomerulus to the another glomerulus. Granule cells make reciprocal synapses with the dendrites of tufted and mitral cells. The mitral and tufted cells normally excites the granule cell by releasing glutamate, and the granule cell, which in turn inhibits the both mitral and tufted cell by releasing GABA, gamma aminobutyric acid. The axons of the mitral and tufted cells it leaves the olfactory bulb and form the olfactory tract, which lies in the olfactory sulcus. So this olfactory tract, it proceeds backward to the olfactory bulb to anterior perforated substance on the base of the brain, where it divides into lateral, medial and intermediate olfactory striae. The axons of the mitral cells, it ends in the lateral olfactory striae. Axons of the mitral cells, it forms the lateral olfactory striae which terminate in olfactory cortex. The axons of the tufted cells, it forms the medial olfactory striae that crosses the midline in the anterior commissure, which reach the contralateral olfactory bulb and synapses with the granule cell. The other fibers, it forms the intermediate olfactory striae that terminate in the olfactory tubercle. Olfactory cortex. Olfactory cortex, which has anterior olfactory nucleus, piriform cortex, amygdala, entorhinal cortex, and neocortex. Anterior olfactory nucleus, it is mainly concerned with coordination of impulses from the contralateral olfactory cortex. Piriform cortex, it is activated by act of sniffing. It is concerned with olfactory discrimination and conscious perception. Amygdala, it is concerned with emotional responses to the olfactory stimuli. Entorhinal cortex, it is mainly concerned with olfactory memories. And neocortex, it is mainly concerned with conscious discrimination of odor. Next, physiology of olfaction. Olfactory receptors, they are stimulated by the odoriferous stimuli. Normally, the act of breathing, that means it is a type of act of sniffing, okay. Through the action of breathing, the olfactory stimuli enters into the nose and passes the impulses to the olfactory mucous membrane. Sniffing, it increases the concentration of the olfactory stimuli. The substance, the olfactory stimuli penetrate the mucous layer and bind with the receptors, encoding of the olfactory information. So after binding the odoriferous substance to the receptors, the odoriferous substance, it activates the G protein and activate adenylase cyclase second messenger system. So because of that, cyclic AMP level increases. That opens cyclic nucleotide gated cation channels. It leads to entry of more positive ion inside and it leads to depolarization. That is called generator potential. That is only local potential changes. So it is called generator potential. When it reaches the threshold level, it generates action potential and it propagated. So in this picture you can see, so odoriferous substance, it binds to the, here you can see the odoriferous odorant molecules or odoriferous substance, it binds to the receptors present in the olfactory mucous membrane, then activate G protein Then it activates the cyclic AMP second messenger system, adenylate cyclase cyclic AMP system. So activation of adenylate cyclase, it split the ATP into cyclic AMP. The cyclic AMP, it causes the opening of the voltage gated calcium channels. The calcium ions enters inside and it leads to depolarization. So initially it generates the local potential changes, that is called generator potential. When this generator potential reaches the threshold level, it generates the action potential. Stimulation of olfactory cells. The same thing, what we have discussed earlier, the same thing given in here also. The processing of information, odoriferous information in the olfactory bulb. The odorant information encoded spatially in the olfactory bulb. The signals are refined by, so all impulses enters into the olfactory bulb. Mixture of impulses enters into the olfactory bulb, where in the olfactory bulb, the signals are refined by lateral inhibition. So lateral inhibition mainly caused by granule cells and periglomerular cells. Signals also refined by multiple input from the olfactory cortex, basal forebrain and midbrain. The sensory information is extensively processed and refined in the olfactory bulb. Next, transmission of odorant information to the olfactory cortex and neocortex. From the olfactory bulb, the information, odorant information is transmitted to the olfactory cortex. From the olfactory cortex, it is transmitted to frontal cortex directly and orbitofrontal cortex indirectly. Omeronasal organ. So omeronasal organ, it is present in the reptiles. It is not well developed in humans. It is not reptiles, rodents. It is very well developed in rodents. This organ is mainly concerned with the perception of odors that act as pheromones. Next, factors influencing olfactory functions. Threshold of the olfactory receptor. So, the threshold of the stimuli. Okay, it's mainly responsible for the perception of the odoriferous substance. Concentration of the odor. So, if more concentration, the perception of the smell is more. Solubility. More soluble substances, it produce more odor. Adaptation. So, odoriferous substance adaptation. So, normally, when we smell repeatedly, adaptation usually occurs. It usually occurs rapidly with a continued exposure. So, adaptation usually not only occur for the agreeable or pleasant odor, it is also occur for disagreeable odor. The mechanism of adaptation are inactivation or desensitization of the receptors due to phosphorylation of the receptor by phosphokinase. Second, adjustment in the sensitivity of cyclic nucleotide gated to cyclic AMP. Role of pain fiber in the nose. The olfactory mucosa also supplied by the trigeminal nerve. So they are stimulated by irritating substances. So they are responsible for sneezing, lacrimation, and other reflex responses. Stimulation of trigeminal nerve is responsible for initiating sneezing, lacrimation, and other reflex responses. Applied aspect, anosmia and hyposmia. Anosmia is a lack of or absence of smell sensation. The causes for the anosmia are injury to the olfactory nerve or olfactory bulb, intracranial lesion or abscess, tumor or meningitis, nasal obstruction, edema or enlarged turbinate, atrophic rhinitis in the old age, parasmia or dysosmia. Parasmia means altered sensation of smell. That means the pleasant odor it perceived as unpleasant, and unpleasant odor it perceived as a pleasant smell. The causes for the parasmia are recovery phase of post-influenzal anosmia, intracranial lesion, and the next topic is measurement of the sense of smell. It is measured both qualitatively and quantitatively. Qualitatively by using uh, qualitative testing, doing in the usually we are doing in the lab by asking the subject to smell the odoriferous substance and to identify that. That is for qualitative testing, and quantitative testing is mainly by the instrument called olfactometer. Next, sense of taste. Taste sensation is one of the very important sensation. The taste sensation, the site of taste sensation is taste receptors located in the taste bud. Taste receptor cells located in the taste buds. The taste buds are located in the papillae, palate, pharynx, epiglottis, and upper third of esophagus. Papillae, there are different types of papillae: circumvallate papillae, fungiform papillae, foliate papillae, and filiform papillae. Circumvallate papillae, which form a V line on the surface of the posterior tongue. Fungiform papillae, which are present over the flat anterior surface of the tongue. Foliate papillae locate in the folds along the posterior lateral surface of the tongue. And filiform papillae are small conical projections, helps in breaking up of the food particles. Taste buds. Taste receptors located in the taste buds. So taste buds, it has the diameter of 50 to 70 micrometer and it is barrel in shape with central pore. So in the picture you can see, the picture shows the structure of the taste buds. It is a barrel shape with a central pore that is called taste pore. The taste buds, it has three types of cells: taste receptor cells, basal cells, and supporting cells. Taste receptor cells are elongated, bipolar shape, and each receptor cells has the lifespan of 10 days. The outer tip of the taste receptor cells, they are arranged around the taste pore, and from the tip, several microvilli protrude outward through the pore. The taste nerve fibers, the taste bud is surrounded by the taste nerve fibers. The taste bud it receives approximately 50 taste nerve fibers. Basal cells, they are present in the bottom of the taste bud and they are stem cells. They will replace the taste receptor cells. Supporting cells are called sustentacular cells. The taste bud, it is innervated by sensory nerve fibers. It comes from the branches of facial nerve, glossopharyngeal and vagus nerve. So, taste buds are supplied mainly by facial, glossopharyngeal and vagus nerve, 7th, 9th and 10th cranial nerve. So, next taste pathway. The taste sensation from the anterior two-third of the tongue, it is mainly carried by the chorda tympani branch of the facial nerve, and from the posterior one-third by the glossopharyngeal nerve, and from the pharynx it is carried by vagus nerve. Taste pathway, it has three orders of neuron. First order neuron mainly by the 7th, 9th and 10th cranial nerve, that is facial, glossopharyngeal and vagus nerve. The taste sensation from the anterior two-third of the tongue, it is carried by the lingual nerve. It is a branch of the chorda tympani. Chorda tympani is a branch of the facial nerve. The taste sensation from the posterior one-third of the tongue is carried by glossopharyngeal nerve. And from the base of the tongue and the pharynx, the taste sensation carried by vagus nerve. These first order neurons, it terminate in the medulla, nucleus tractus solitarius of the medulla. Nucleus tractus solitarius, it acts as a second order neuron. The fibers of the nucleus tractus solitarius, it crosses the midline and join the medial lemniscus. Then terminate in the ventral posteromedial nucleus of the thalamus. This ventral posteromedial nucleus of the thalamus, it forms the third order neuron. The fibers of the ventral posteromedial nucleus of the thalamus, it ascend upwards and terminate in the lower tip of the post-central gyrus in the parietal cerebral cortex. So here you can see the sensation, the taste sensation from the tongue. It is carried by 7th, 9th and 10th cranial nerve, and it reaches the nucleus tractus solitarius of the medulla. This nucleus tractus solitarius, it acts as a second order neuron. The fibers from the second order neuron, it crosses the midline and joined as a medial lemniscus and ascend upwards and terminate in the ventral posterolateral nucleus of the thalamus. This ventral posterolateral nucleus, it forms the third order neuron. The fibers of the third order neuron, it ascend upwards and terminate in the taste area of the cerebral cortex. Next, physiology of taste. The taste buds containing taste receptors are concerned with the perception of taste sensation. There are 5 primary taste sensations: sweet, salt, sour, bitter, and umami. The taste receptors for the sweet sensation, it mainly located in the tip of the tongue. The taste receptors responsible for the salt sensation are located in the front half of the side of the tongue. The taste receptors that is responsible for the sour taste, it is located in the posterior half of the side of the tongue. The receptor for the bitter taste sensation, it is mainly located in the base of the tongue. Umami, it is a new sensation, taste sensation. It is produced by glutamate. Newly invented taste sensation, mainly produced by glutamate, monosodium glutamate. Transduction of the taste stimuli. The gustatory stimuli, that means taste stimuli, it is converted into electrical signal, that is called transduction of the taste stimuli. So normally the taste receptors are chemoreceptors. The dissolved substances, when we are taking food, that taste substances dissolve in the saliva. The dissolved substances act on the microvilli of the taste receptors. This leads to depolarization of the taste receptor cells, either directly or by through the second messenger. So first it generates the receptor potential. Then after that it is developed as a action potential in the sensory nerves. So once action potential is generated, it transmitted to the cortex. The sensory signals carried by the sensory fibers and travel through the different nerves and reaches the cortex. So in the cortex, encoding of the gustatory stimuli occur. Most of the receptor cell is responsible to more than one stimuli. The afferent neuron receives information regarding a variety of taste stimuli. So it fires optimally for only one stimuli. Neurons in the gustatory cortex are stimulated in response to the pattern of action potential in the afferent neuron. So afferent neuron, it receives information regarding the variety of taste stimuli, but it fires optimally for only one stimuli. Okay, so neurons in the gustatory cortex, the cortex are stimulated in response to the pattern of action potential that is generated in the afferent neuron. Taste threshold and intensity discrimination. Taste threshold, it is a minimum stimuli. Here, taste threshold, so minimum concentration of taste substances which require to produce taste sensation. Okay, that is called taste threshold. Intensity discrimination. So it is nothing but ability of the humans to discriminate differences in the intensity of the taste substance. So for the intensity discrimination, at 50% change in the concentration is necessary. Change in the concentration of the taste substance is necessary. Factors influencing taste sensation. Age. So in the old age, the number of taste receptors decreases. Genders in the female, taste sensation more. Concentration of tastings. So more concentrated substance, it produce more sensation. So it is area of stimulation. Area of stimulation also directly proportional to the taste sensation. So temperature, it increases the taste sensation. So high temperature, it increases the taste sensation, and cold temperature, it decreases the taste sensation. Duration of stimulation. So how long we are uh, we are exposing to the taste stimuli. So that is also one of the factors that influencing the taste sensation. The interaction between the taste producing substances, that is also plays important role in the perception of the taste sensation. Ageusia, absence of taste sensation. So lesions of mandibular divisions of trigeminal nerve, facial nerve, and glossopharyngeal nerve. Drugs like captopril and penicillamine that also produces ageusia. Hypogeusia, it is diminished taste sensation. Dysgeusia, it is disturbed taste sensation. The selective taste blindness. Taste blindness, it is tested by using a substance called phenylthiocarbamide. So we have discussed about smell and taste sensation. So smell sensation, it is mainly perceived by the receptor present in the olfactory mucous membrane, and taste sensation by the receptor cells present in the taste bud. Once again, I'll refresh. The olfactory mucous membrane has three types of cells: receptor cells, tentacular cells, and basal cells. Receptor cells has thick dendrite that is called olfactory rod. It has many cilia and it receives the taste sensation, and the axons of the olfactory receptor cells, it pierces the cribriform plate of ethmoid bone and enters into the olfactory bulb, where it snaps with the dendrites of mitral and tufted cells. The axons of the mitral and tufted cells, uh, the mitral and tufted cells forms the second order neuron. So the axons forms the olfactory tract and it terminates in the olfactory. It runs, okay, once again. The olfactory tract, it proceeds backward from the olfactory bulb to the anterior perforated substance, where it divides into three striae, lateral, medial, and intermediate striae. Lateral striae formed by the mitral cells, and medial striae it is formed by the tufted cells, and the intermediate striae, olfactory striae, it is formed by the other fibers. The lateral olfactory striae terminate in the olfactory cortex. Medial olfactory striae it terminate in the it crosses the midline and reaches the contralateral olfactory bulb where it snaps with the granule cells and other fibers. It terminates in the olfactory tubercle. So olfactory cortex, it is nothing but it is a group of, it comprises of group of nucleus, anterior olfactory nucleus, piriform cortex, amygdala, entorhinal cortex and neocortex. The next one, taste pathway. So taste pathway, okay, from the anterior to three orders of neuron, first order neuron, second order neuron, and third order neuron. From the anterior two-third of the tongue, the taste sensation carried by seventh cranial nerve, that is facial nerve, and posterior from the posterior one-third by ninth cranial nerve, and by the base of the tongue and the from the base of the tongue and the pharynx by the vagus nerve. Okay, these three nerves forms the first order neuron. So these neurons terminate in the nucleus tractus solitarius. The fibers from the nucleus tractus solitarius, it crosses the midline to join the medial lemniscus and it ascend upwards through the medial lemniscus and terminate in the ventral posterior medial nucleus of the thalamus. Last time I said lateral, sorry. It is ventral posterior medial nucleus of the thalamus. So the fibers from the ventral posterior medial nucleus, it terminate in the lower tip of the postcentral gyrus in the parietal cerebral cortex. That is not ventral posterior lateral. This is ventral posterior medial nucleus of the thalamus. Thank you.